Hello, my name is Bradley Alisea, and today I'm going to be presenting on Embryo Networks as generative divergent integration. This is a talk for Networks 2021, and I'm presenting this work on behalf of the DevaWorm group and my collaborator Daniela Cialfi, and there are our Twitter handles. So today I'm going to talk about these divergent networks, but one part of these divergent networks are embryo networks. And so embryo networks are, and you can see with the diagram on the left, these blue circles are cells that emerge in the embryo, and the black arrows are bidirectional connections between the cells. So these embryo networks might represent interactomes or molecular signaling between cells. And so we can think of these embryo networks as three-dimensional constructs that exist in 3D space, and that 3D space is roughly congruent with the anatomical axes of the embryo. And then we can think of this in terms of time and context. So the time is the developmental time at which cells are in the embryo, dividing and differentiating, and then the context can be things like the angles of cell division or other features that we might want to add into the model. And we have a Jupyter notebook on GitHub that contains a lot of the details for that. This leads us to this five-dimensional structure, or this five-dimensional data structure in which we kind of view the embryo in the DevaWorm group. So on the left is a bunch of cell tracking data. It's a point cloud where you have data from like 300 and some embryos that are, the different cells have different identities, but these cells are tracked in different embryos over time. And the idea is we can take these data, average them out for certain cell identities, and find the location in space. And so these three axes are X, Y, Z. And then on the right, we have these lineage trees that, and we work in C. elegans. So this is all very uniform across embryos and across lineage trees. So on the right, we have a lineage tree that represents time over time. So there's cell divisions over time. And then there's this parameter I, which is sort of the axial organization of the lineage tree, which we can interpret as identity and other, cell identity and other things. So we have a lot of information here that we can map to this network. And so cell division provides a number of challenges for looking at this in a network, from a network perspective. The first is that the graph diameter will expand or grow in the number of nodes over time. So as cells divide on orders of 2, this network will grow, both in size, but more importantly in its density. And so this is the consequence of this is that local connectivity increases over time. The nodal density will increase, and especially as you get differentiation and you get migration of cells, certain areas will tend to take on, modular form. And so global modularity thus increases. And this is due to differentiation events in what we call bifurcations. And so bifurcations are essentially cell divisions, but in this case, you can see that you have these cells that start out as a single cell, and then they start to expand into something that looks like it's going to divide, and then finally you have this bipartite structure, this bifurcation, where you now have 2 cells, where you had 1. And so this bifurcation structure, this leads to the questions, how many parts does the network fragment into over time? So how many nodes do we fragment into, but does this lead to higher order structures? And so we can look at this. In terms of spatial connectivity, but also temporal connectivity. So in this example of the interactome, you have this network structure in anatomical space, and you have different colored connections. And these nodes don't all exist at the same point in time. These have been plotted to show that you have different networks at different points in time, and the connectivity changes as you have different levels of cell division. So you can see here we have a hairball network that shows what the embryo network will look like over time in different, with the anatomical orientation here. P1 is the posterior end, AB is the anterior end. And we can also then connect this to the connectome, the neural connectome, because we know, for example, the locations of all the cells in the developing embryo, but we also know their identity. And so that allows us to look at neural differentiation over time and the emergence of this connectome. So this is what our generative divergent integration is referring to, taking these embryo networks, turning them into sort of these hybrid embryo networks and connectomes, and then looking at how those two things evolve over time. And so here you can see an example of the connectome that's emerging in development. This is at about 300 minutes of development, of embryonic development. And you can see that you can track the different neural identities based on their lineage tree position. So you can see that for different parts of the lineage tree, we can find neurons, we can find their connectivity, and then we can link that back to the embryo network. So when connectome neurons emerge in the embryo, we find that they're actually very conserved relative to the adult. So in this case, we're going from this point cloud, we're creating an embryo network, and then we're trying to find within the embryo network, or within a sort of a refined point cloud, where those neurons are located. And you can see that they have a certain axial distribution in the embryo, and so we can integrate those two networks together, and it's very predictable across individuals. And so again, you can see this in three-dimensional space, you can see this in two-dimensional space, and then at the bottom here you can see this in the adult, where these neurons are located within the organism. And so you have two networks here. You have the connectome, which is all these dots here, these bright dots, and then you have every cell in this organism, which also forms another network. So this is an example of generative divergent integration. And we can look at the 8-cell example and the 24-cell example. So in the 8-cell example, we can look at only the embryo network because the neurons haven't differentiated yet. And we can see that we can set a threshold of a certain distance between the cells, or the cell centroids, to give us, either two cells are connected or two cells are not connected. So if they're close enough together, the cells are connected. But if they're farther apart, they're not connected. So you can see that the cell up towards the anterior end and the cell at the posterior end are not connected. But they could be indirectly connected if there are enough cells intermediate to it. And they would be indirectly connected by a certain order. But you can see here the 8-cell embryo. In this case, and this is not empirical, it's just something I drew, it's a bipartite network. It's not a network that's integrated in any way. It has little pieces that are it's fragmented essentially. So we say for all developmental cells blue, the distance t between cell centroids, and that's how we get our connectivity. And there's sparse connectivity due to the larger cell size and sort of the sparseness of this embryo. By contrast, we go to this 24-cell example. And again, this is just a cartoon, but we get actually at this point, we're getting two different types of cell. We're getting the somatic cells, which are in blue, which we see in the 8-cell, and we get these light green cells, which are neurons or emerging neurons. And so now we can make the connection between these two types of networks. We have these connectome or neural networks that are starting to form, and then we have these embryo networks, which are already becoming more dense and connecting together. There's no longer this fragmentation of the network. It's totally joined together, but you can see that the neural network and the somatic network are joined together by the same criterion. And so we actually have this dual network. And so all neurons that are green might share gap junctions and have a special sort of, and those networks are at this point fragmented, but they share properties that the somatic cells do not. Nevertheless, you can say that they're connected because we have this criterion of distance, which is of course important in signaling and other things. And so we can draw this network in a number of ways, but the idea is that they're integrated within one another. They're embedded within one another. And so, then we have a larger question, which is that in terms of developmental spatial connectivity and connectomes, what kind of network is it? Is it a random network? Is it a scale-free network, or is it a small world network? And these are data from an adult zebrafish. So the adult zebrafish exhibits this type of connectivity, and in its neural network. But the question is what do we expect for developmental networks? And so this is vexing because it's not necessarily going to yield these types of patterns, because it not only is it not static, but it also has certain properties that maybe we need to figure out if there's maybe a new rule or a new set of rules that we need to consider for this. So we have a number of candidates for this. We have the New World Networks, which are small world networks with expansion. We have chimeric states, which are simultaneously coherent and incoherent networks, which is maybe more relevant to our divergent generative networks. And we have small world networks, but constrained by spatiotemporal sampling. And since we have a spatiotemporal component here, that might be useful. We have network connectivity preferences that influence later activity in ways that affect symmetry. So network connectivity is having an effect later on. And this is, of course, relevant when you have a number of cells growing in the embryo. And then finally, you have these generalized hierarchical signatures. So where you have hierarchy in the network, or when you begin to have hierarchy, you begin to see it, and it has its own network dynamics. But why don't we focus in on New World embryo networks and consider, what their properties are, because the New World embryo networks are the ones that allow you to grow nodes and consider, how those things are connected into the old or the antecedent network topology. So in terms of New World embryo networks, these are multi-level networks. We have a number of proximity and adjacency measurements, such as convex Hall measurements over time. And we can understand that using topological data analysis. We can also use differential network diameter and look at the network diameter between T1 and Tn, whatever that time is, so we can get an accounting of how these things grow over time. We can also measure expansion rate in different ways. So we can look at differential path length ratios. So between time 1 versus time n, we can look at the difference between the path lengths. Or we can look at differential clustering, again, between time 1 and time n, so we can see how much clustering is gained over time. We assume that we start out with a random initial condition that doesn't have much structure. So the clustering will, of course, increase, but by how much? We can also look for new types of topologies, and there are at least 3 candidates for this. One are feature-rich networks. So these are topological features that capture emerging tissues, fluid dynamics, and gene expression cascades. A second one are multiple worlds. So multiple worlds consists of different processes and structures captured in an n-partite network with weak connectors. So this is more relevant to combining the connectome and the embryo network into one single system, but also something that has differential functionality. And then finally, semi-integrated networks, which of course are interrelated phenotypic modules and functional systems. So this is like the brain and the body problem, but this is actually something that we can quantify in our networks. So we've come up with one thing for now that might be a candidate, and it's something we're proposing called the density bifurcation model. So the process of increasing connectivity and development occurs as follows. At first, you get cells that divide and migrate. And as a result of that, based on our sort of criterion, the connectivity increases. Secondly, cell migration enriches local communities and cliques, so you get this enrichment of structure. Thirdly, the function of cells diverge over time, so there's this differentiation function. Not only do you get connectomes and embryo networks, but you get structure within embryo networks. You get different organ systems and things that emerge. And so at least 2 interconnected networks emerge from this, but probably more as we go through development. Finally, interconnected networks provide weak ties, and these are functional interdependencies between emerging tissues. So these interconnections between different types of networks, these are actually weak ties. So if we go back to our cartoon, we can see that the neural networks and the somatic networks are connected, but these are weak ties that might, say, not exhibit all of the features of 2 cells that are connected within the somatic network, but nevertheless have some influence. And we use the term weak tie because weak ties are, they'd be more loosely associated, but still associated and have topological effects. So future directions for this work. we want to capture embryo dynamics more effectively. I showed an image of a hairball network. I showed images of where you can look at multiple layers of the lineage tree simultaneously, but we really want to get a better accounting of the embryo dynamics. And that means looking at time series and building networks that are dynamic in time and multi-level in time. So here you see examples of embryos that are evolving over time, that are changing their morphology over time. And this is the sort of thing we want to pick up on. We want to be able to identify a lot of these movements and maybe what we call meta features, which we're currently kind of beginning to understand with using machine learning and deep learning, where you get different movements of the furrow and different movements, things that emerge as cells differentiate and move around. And so those are the things we want to characterize with networks. So thank you for your attention, and thanks to the DevaWorm group once again and the OpenWorm Foundation.